The feedback video for the September interim assessment. I'm just going to go through some skills and common mistakes. If you want answers to specific questions, the mark scheme is at the back of this presentation, so just fast forward. This table caused some issues in that these have to be comparative statements. So, for example, if you put helical for starch, you'd have to put straight for cellulose because they're comparing the shape of the molecule. For example, if you put no microfibrils for this, you put microfibrils for this box here. If they don't form comparative statements, you can't gain any marks. The other thing to watch out for is the list rule. It's asked for 2 statements. So if some students put other ones underneath, you're at risk of losing marks. So beware of the list rule. This bottom question here was asking about cellulose's function. So you need to state what it is, that it's strong, the idea that it helps strengthen the cell walls to prevent osmotic lysis or bursting when the plant cells are turgid. And then one way in which the structure of cellulose links into that function, the idea that the parallel chains are held in place by hydrogen bonds, or you could say this forms a microfibril, because that is what makes it strong. You should be able to identify any of these bonds. Some students struggled, so let's start with probably the easiest one to spot, which is letter C. So hopefully you recognise that this is a glycosidic bond between 2 monosaccharides. Letter B, hopefully you can see it should be a very recognisable one, that's a peptide bond. You've got your C double bond O and your NH joined together, so that's a peptide bond. This one's harder to spot. This is one of the other main biological molecules. You've got an ester bond here. Okay, so this C double bond O is an ester bond. So that would be found in triglycerides, where the fatty acid has joined onto the glycerol, which would then carry on down there, and you'd have 2 other fatty acids there. This one here is between 2 amino acids, and that is a disulfide bond. So let's look at the question. On the whole, that was absolutely fine. This one here is quite interesting. May occur in the tertiary, but not in the primary structure of a protein. So an error some students were making was put in the peptide bond. That cannot be the case because this would be found in the primary structure as well, joining every amino acid together. So that one would have to be the disulfide bridge, only found in the tertiary, not in the primary. This last one here, most students gained 1 out of 2 marks. The reason being, students failed to put the idea that a saturated fatty acid, yes, only contains single carbon-to-carbon bonds, but it acts in the main part of the hydrocarbon chain, ignoring the carboxyl group, where you've got C bonded to C bonded to C in the R part of the fatty acid, the hydrocarbon chain. This was poorly answered. So first of all, a description question. Describe how the concentration of omega-3 fatty acids changes over time. So, a description. So, hopefully you can see that the line decreases, but it doesn't decrease in a linear fashion. It doesn't go straight down. So we need to describe the idea that the drop is steeper to start off with, and then it levels off after 140 days. So steep decline at first, and after 140 days, the omega-3 fatty acids levels off. This question here was also poorly answered. It's a rate of decrease. So a rate is the change in y divided by the change in x, where x is time. So we need the change in omega-3 over 40 days divided by the time, which is 40 days. So times 0, we had a value of 3. Times 40, we had a value of 1.3. 3 minus 1.3 is 1.7. Divide that by the time taken, which is 40. So we should have an answer. 1.7 divided by 40 gives an answer of 0.043. You might be wondering why you don't put 0.0425, which a lot of people did, and it's to do with significant figures. The vast majority of the answers, not excluding that one, these are all to 2 significant figures. So you could do your answer to 2 significant figures. Really, you should use the lowest significant figure that you have. So this one's only done to 1 significant figure. So you could also have 0.04 as an answer. Part 2 here, extremely common question. So why is it expressed as a percentage of total fat? If you ever get a question like this, it's due to, so you can compare the results. It allows comparison. So if you didn't do that, in this case, for the second mark, always look at what your dependent variable is. So we're measuring percentage of total fat in the milk. So that would mean that there's variation from cow to cow in the proportion of omega-3 compared to other fats in the milk. So you have to make it as a percentage to allow comparison. Valuate questions are perhaps the hardest ones to answer. Whenever you tackle this, you should be putting points for and against. So points for there being a link and points against being a link in this case. So the points for there being a link is the idea that omega-3 does decrease over time or after being fed on corn. However, with any link, it's only a link. We can't prove that that is the only factor causing the results. There might be other factors. And we cannot prove that it was caused to the decrease was due to corn because we've not used a control group. So you cannot make any definitive link there. We don't have a control group. It's also in one study, so it might not be representative. So they're common things that you need, common terms that you need to use in an evaluation question. Have they used a control group? Yes or no. Have they done a large enough sample size? If yes, it's representative. If not, then it's not. Well, not a lot, but the biggest error with this question was not lack of detail in answering the question. It's about how a change in the primary structure may result in a different three-dimensional structure. So if you don't say there's a change in the order of amino acids, which results in bonds forming in different places, which means that you have a different tertiary structure, if you don't use the words difference, then you don't get any marks. Another way that students lost marks is that they used primary structure. So they thought they were getting the first mark saying a change in primary structure will cause. But you can actually define what primary structure is really. So replace that with a change in the order of amino acids. That will get you your mark. You've told the examiner you know what a primary structure is. So here we've got phenylalanine, and here we have an addition of OH, which means hydroxylating. This was a tricky question, so this would be one that's aiming for trip a lot of students up, see if you understand what's going on. So tyrosine can be made by hydroxylating phenylalanine. So let's have a look at the diagram to see what that is. Very common mistake by students happen here when you join two monosaccharides together. It doesn't matter what the monosaccharides are, you have to use adjacent hydroxyl groups. So here we've got two adjacent hydroxyl groups. You take off one completely and you take the hydrogen from the other. There you've got your oxygen, which can bond to this monosaccharide. You could have also used this hydroxyl group and this hydroxyl group. The biggest mistake that people make is using a hydrogen with a hydroxyl group. Yes, that will, when you remove it, will form water, but you are not left with an oxygen to form the link. If counting the number of carbons in fructose, every time that the angle changes direction, you've got a carbon. So you've got 4 carbons in the main part of the chain, and you've got 2 in the groups branching off. So you have a total of 6 carbons. This is a fairly tricky maths question. So it's asking for the mass of cellulose in 1 kilogram of grass. So first of all, you should realise that there are 1,000 grams, because the final answer wants to be in grams. So let's convert that into 1,000 grams to start off with. 80% of the grass is water. That means 20% contains the carbohydrates. So what mass of carbohydrates have we got? So 20% of 1,000 is 200 grams. So we've got 200 grams of carbohydrates. Of that 200 grams, 25% is cellulose. So cellulose is 25%. 25% of 200 is 50 grams.